The Farallon Islands are located off the coast of San Francisco, California. Placed as a National Wildlife Refuge in 1909, the islands are federally protected and are off limits to the public. The waters are rich in nutrients and the islands attract an abundance of marine life. In fact, the Farallons support the largest nesting seabird colony in the contiguous United States. All is not as pristine and undisturbed as one would hope, though, when it comes to the Farallons. Due to past human travels, the islands are home to invasive house mice, and the Farallons has some of the highest mouse densities ever recorded at over 500 per acre. Sadly, this is not a unique situation, as 80% of the world's islands harbor invasive rodents. Rodents pose significant problems on islands. For example, in the absence of humans, house mice are no longer commensal, and their diets on islands shift to eating native plants and invertebrates. The worst of these impacts, though, has been to native seabirds, as house mice on islands are known to consume seabird eggs and chicks. In the fall of 2013, I personally had the chance opportunity to go and collect wild mice from the Farallons and take them back to North Carolina State University. One reason for why we collected the Farallon mice is because little is known about how wild mice on islands behave compared to their laboratory counterparts. A large part of my research focuses on the behavioral and reproductive differences between Farallon mice and laboratory mice. For instance, we know that wild mice have faster velocities, can jump, and build elaborate nests. And we know that the Farallon mice will breed and produce litters with laboratory mice. Current work is now looking at when wild females are given a choice, who does she prefer to mate with, a Farallon male or a lab male? The other reason we chose to study mice from the Farallon Islands is to look at a new method for pest control. Typically, islands, including the Farallons, try to remove invasive rodents through poisons. This method requires tons of poisons to be dropped on the islands, raises human and animal welfare concerns, and doesn't always work for mice. Instead, an alternative is being explored that uses gene drives to remove invasive mice from islands. A gene drive is a gene that drives itself into the population. In other words, rather than the standard 50% chance of being transmitted, these genes are inherited nearly all of the time. Mice have a naturally occurring gene drive called the T haplotype. Think of it like super sperm. Those sperms who have the drive are preferentially inherited. One way you could use this drive would be to sex bias the population, making it mostly male. If the population doesn't have enough females, it won't be able to survive. In mammals, the SRY gene is the male determining gene. This gene is all that is required for a mouse to be phenotypically male. If one was to put the SRY male gene into the T haplotype, then all of the mice that inherited the drive would be male. Now, gene drives are not without societal concerns and risks. There are questions about what if the mouse mated with another closely related species or reached the mainland. There is also the question of how would you even develop a gene drive mouse that can survive on an island? This latter question has been the primary focus of my research. I made wild and lab mice to create hybrid mice. In doing this, I see no significant difference in litter sizes compared to the wild mice, and hybrids may even be stronger competitors. Conservation on islands is crucial, as islands, while only 5% of the Earth's landmass, contain 40% of endangered species and have a disproportionate number of extinctions. While the gene drive technique is not currently being utilized for the Farallon Islands, it is something to consider. In summary, conservation issues involving invasive house mice may prompt new ways of thinking about conservation.